Okay, so this is going to be our last lecture video for the semester. Your enormous nightmare of having to listen to me say "all right" at the beginning of every video is almost over. Don't worry. Okay, so last time I was talking about questionable research practices and how those can influence the findings that come out in actual studies. Hope it's clear that those are relatively pervasive and that they actually are pretty big problems that we need to deal with as a field and as scientists more generally. So what can we do to reduce the occurrence and influence of questionable research practices? There are a number of things we can do that most people would say start with disclosures. Disclosure, it kind of sounds like a big legalistic thing, and it sounds like a burdensome thing for scientists to do. But that is really the wrong way to think about it. Science is about transparency. If people don't know what you did to obtain your results, then how can they have any confidence about what those results are? So transparency and being willing to disclose things are really the hallmark of, in a lot of ways, good, rigorous science. That you are not being shady, basically, about the things that you're doing to obtain your results. And I understand that sometimes there are going to be legal reasons that people can't disclose various things, or confidentiality or privacy reasons. But for the most part, people should be willing to disclose what they can. So in general, people should be willing to tell you exactly what they did. And I know that I talked about that in terms of what your method section should actually include in class. And I know Jenna talked about that too, that from your method section, people should be able to replicate your study completely. And of course, I've also talked about the word limits in publications. So those things are kind of countervailing pressures, right? That you want to include as much as possible, but you don't want to waste all your word space talking about the number of items that you had in a particular, well, I guess that's not a burdensome thing, but going into enormous detail about your methods such that someone could actually replicate it. Happily, we are in an age where page numbers and space are basically meaningless in a lot of ways, in the sense that you can upload supplementary electronic files to any article anymore. Supplementary appendices are the norm now, and really one way people should be using those is to include all of their materials for every study that they run in every article that they try to get published. Or even beyond that, every project that you're involved in, you should post these materials up so other people can use them and know exactly what you did. Same with your the syntax, the code that you use to clean your data and run your analyses. You should include those things so people can know exactly how you did what you did and correct it if necessary, and help you understand how to interpret your results too. So that's disclosures in kind of a general sense, but I want to talk about some other more specific types of disclosures, and one specific thing that you've already heard a little bit about. Doesn't, I guess, totally fit into disclosures, but still a useful thing to think about. Pre-registration is something that Daniel Simons talked about in the video that you hopefully have watched last Friday or last Monday. But it has to do with being upfront about what you predicted and proving that you predicted what you predicted by pre-registering it. There are lots of new places that people can do this. One of the more popular is called the Open Science Framework, osf.com or osf.io. I think osf.com actually just redirects you to osf.io. But there, researchers can make profiles where they can list all the different projects that they're involved in, and that's somewhere that they can share, their materials, their results, share their data if they are able to. But it's also a place where you can pre-register your hypotheses. So if people, I mentioned in the last presentation, one of the questionable research practices, hypothesizing after results are known, harking. This is something that we all recognize as being an unreasonable thing to do that inflates the false positive rate, but I think you would be hard-pressed to find anyone that has never done it, and I think in the 60s, 70s, 80s, 90s, 2000s, and even some people today, but definitely in those last decades, it was the norm. It was that you ran a study, you included as many dependent variables as you could think of, so you weren't wasting your time. You manipulated things in as many ways as were practicable with your sample size, and you just found whatever results you got, and you published whatever you got saying that you predicted it. That was how people were taught in graduate school to run studies. So, I mean, your false positive rate, you're expecting 5% of your results to just be significant due to chance, right? Well, if you collect 20 versions of your dependent variable, then one of them is going to be significant. And you can't just go report that, even though, people in the past might have been taught that that's how you do science. But we know now that that is not how you do it, and it leads to misunderstandings about the types of relationships that are actually going on between variables. So to combat those types of things, you can pre-register your hypotheses. You can lay out exactly what you predict, exactly what would qualify in your eyes as support or non-support for that prediction, and then there's no ambiguity later on. There's no you looking at your results later and saying, well, does this support it, or rewording your hypothesis or anything like that. You've made a publicly visible commitment to that hypothesis. You've said what you're going to take as support for it, and that is all out there. And violating that would be an enormous taboo, and would be something that would, be kind of a mark on you for other studies that you'd run in the future. So again, this is really, sorry, as I yawn, this is really important stuff. If you haven't guessed at this point, I'm recording a lot of these at night because my children are asleep and thus not awake and able to interrupt them with sounds that they make. And there's my big non-reveal at the end that I am tired doing this at night. Anyway, pre-registration. Not only can you do this with hypotheses, but you can report and pre-register lots of other things, like how you're going to trim outliers, what types of exclusion rules that you have based on like demographic characteristics, or if people fail attention checks, or if they have a certain pattern of responses that you think is unreasonable or not applicable to your study. You can register those, and you can register the types of analyses that you plan to run. So it's also the case that in addition to just like saying they hypothesize things, people will just run tests in a bunch of different ways, and then they'll just report to the one that works. And that, again, is inflating that false positive rate by doing multiple tests. So if you pre-register your analytic strategy, then you have made a commitment up front. I'm going to test my hypothesis in this way. And then there's no ambiguity. You do it the way that you said, and you are holding yourself to that commitment, and other people can see it publicly. A nice thing about OSF and other places you can pre-register, you can embargo registrations. So you can say like, I'm going to make this and I'm going to save it on this website. It's unchangeable, not deletable, but I don't want it to be visible for a year. in case you like don't want people to scoop you, take your idea or something like that, or ask you about things you're not ready to talk about publicly as a researcher. So you can embargo things up to, I believe, 2 years, which is a nice feature of these. So people with that complaint, it's not valid. You still should do this. Sharing your data is another disclosure thing that is, again, kind of tricky, because sometimes we have, identifiers of participants, or we have medical data, or something that's otherwise private or confidential. So obviously in those cases you can't share that data, but there are frequently ways that you can clean it or augment it in a way that doesn't violate someone's privacy or endanger anyone. But sharing your data, again, used to be kind of a weird taboo thing that like you didn't want people to see it, but now we are better at recognizing that you should not be the only person dealing with it. If for nothing else, in those fraud cases, that like people should be able to look at others' data in order to kind of check their work. And more often than not, people aren't really, checking. It's just kind of a trust thing that you allow your data to be out there to prove to people that you are willing to let other people look at it and play with it. And then, we can be more confident that you are not lying to everyone and pulling one over on people. And these types of transparency, they just make people work harder. They make people try to be more rigorous. They make people make better decisions because there's accountability. And we know that as self-interested people, when there's no accountability and there's ambiguity about rules, then we'll do things that are good for us and bad for everybody else, including the field in general and the state of our knowledge as a human race. But when we have accountability, we can be more confident that people are making the right decisions and that we ourselves are going to be capable of making the right decisions. So one last thing kind of related to that runs against that. It's hard to be a good scientist and keep your scruples about you and make sure you're doing things correctly. But if you don't find the result you expect, that's just what the result is. And that's how science works. And being a responsible scientist is about reporting things as you observe them and not bringing your biases or your incentives or your problems to bear on it. And again, like that can be very difficult. And it's just a responsibility that you accept when you start doing science. It is, it's not a perfect system because human beings are involved. We introduce a lot of selfishness and chaos into the things that we do, but in order for science to be self-correcting and for it to work properly, we need to be able to trust people and trust ourselves to be able to do these sorts of things and let science work in the proper way. Probably the biggest organization, or definitely the biggest organization that is working to help these types of endeavors is the Center for Open Science. This is a nonprofit organization started by Brian Nosek, who is a social psychologist who studies implicit attitudes and politics a little bit, and Jeff Spies, another researcher. They founded this to meet all these goals and facilitate people's work in these areas. They have that website I mentioned, the OSF, the Open Science Framework. This is that place where you can make a profile, post your pre-registrations, your data, your syntax for your code, whatever you want to post, and it even helps with workflow a little bit, so it gives people an incentive to do it for that reason. It's very popular. If you some of you move on to like grad school and stuff, and you're looking for people's papers or things that they're working on, look up people that you might be applying to work with in grad school, or people that you can't find papers for. You might be able to find their data and find papers and stuff on there, and learn a little bit more that way. So it's free, it's open source, it's flexible. There's a lot of really interesting stuff you can do with it. And one cool thing too, it is a nonprofit. But people of Brian in particular, his stature and expertise, they get paid enormous, like sickeningly enormous speaking fees, to talk about their work. And Brian, some of his work is talking about open science stuff. And I know that sometimes those fees are in the tens of thousands, right? They're getting $25,000 to $30,000 if you're like a famous scientist that talks about their work. But I know that Brian, whenever he talks about open science stuff, which is very often, he gives all the money from that to the Center for Open Science. So a good chunk of their funding comes from him, going out there and taking the time to educate people about how to do science better, and then feeding that money back into the effort. So I've always thought that was a pretty cool thing for him to do. But then, he also gets to go talk about implicit attitudes and get, $25,000 a pop for doing that. So, he's doing okay. The Center for Open Science also, they fund and support a lot of other endeavors that have to do with open science, like the many labs projects and reproducibility projects. I'll talk about those, I guess, in just a minute, but many labs projects is a way to get a lot of different labs, as you might expect from the name, involved in testing and replicating different questions. So I'll talk about a couple of those efforts in just a minute. And the reproducibility project is pretty similar, but trying to estimate effects and see if different things are replicable on like a slightly different scale. The many labs project is usually lots of labs trying to do the same thing, testing the same thing. The reproducibility project is a little bit more varied. I want to make a distinction here before I talk more about those, though, between reproducibility and replicability. Reproducibility just means that you could redo a study. You could redo the method. You could redo their same exact type of analysis. So reproducibility is, facilitated by open materials and open methods and things like that. Reproducibility just means that you are being open and transparent enough that your study could be reproduced by somebody else based on the information and resources and materials that they have. Replicability is different. This is whether a finding is repeated. Not a study is repeated, but the actual finding. So is a study that's studying the same thing as another study finding the same result? So if study A about the connection between aggression and violence finds a particular relationship, does another study find exactly that same relationship between those two variables? And there are different kinds of replications that you can run. Exact replications are using all of these same exact materials from somebody else and trying to do it literally exactly the way they did it. Same instructions, same measurements, same analysis, everything's the same. Conceptual replications are a little bit different. They're kind of like extending and making analogies of different findings. Like if you are priming somebody with words about the elderly, I talked about that earlier, and seeing if that activates people's concept of slowness and maybe they walk slower. Okay, well, maybe a conceptual replication of that is that you prime elderly words and you see if people perform a different task more slowly, or if they shift their perceptions in a slow manner or something. So a conceptual replication is different because you might operationalize your dependent variables or your independent variables differently and show that this relationship exists even when you don't do things exactly. So these things are really complementary. We need exact replications to show that, to try to zero in on an effect size, but we need conceptual replications to show that the findings are not just an artifact of this exact method. So these things really go hand in hand and are really necessary. And again, I would want to just emphasize that replicability and I mean not replicability, but replication and the endeavor of replicating things, it is not done nearly enough. It's what science hinges on, that we are replicating each other, but nobody wants to replicate because there's no incentives there. There's no journal of replications. There's no journal of null findings that we can submit things to when stuff doesn't work out. And people want to hang their hat on an idea that's theirs, not use somebody else's. So we need massive changes in the incentive structures to be able to promote and encourage replication, so people do it more often. One way that people have been encouraging replications is through this mini labs project. So this was really a way to get people that maybe don't have the time or funding to do their own studies and develop their own studies, to still be involved in research. So they picked studies that could be done relatively easily, studies that we kind of know replicate already, so we can kind of get a baseline. Our studies that we know are going to replicate, going to actually still replicate, and things that we maybe don't know. So replicating further some other things to understand how well this works, and then some unknown studies to test the replication of those. So the first Many Labs project took these relatively well-known things, some of them like anchoring and adjustment heuristics, gambler's fallacy, and some unknown things like sex differences between implicit math attitudes, and the effect of imagined context on prejudice reduction. So these types of plots, you may or may not be very familiar with them, but these are like looking at effect sizes, basically. Standard mean differences, we talked about Cohen's D, that's what you're looking at, is these differences between conditions across all these different studies. So this is an effect size of 0 on the line. Something negative would be a negative mean difference, and anything higher is a relatively big effect. So 1 is usually that means 1 standard deviation in difference, and anything bigger than that is a pretty large effect. So we're showing these big effects for some of these classic studies, and as we get down to some of the ones that are a little less well known or maybe a little newer, we're seeing smaller effects, or we're seeing these average effect sizes in the middle here that are at 0. So maybe currency priming, maybe that effect is not real. That's what this would seem to indicate, that all these different dots are different labs that replicated this, and they're all kind of clustering around 0. So we may have some skepticism about whether currency priming actually is anything, right? Even though the original effect, this X, was close to a pretty big effect size of 1 here, all the replications are showing that it's not. So again, these are just another 20 labs or something, but they're all converging on an estimate here that it really isn't existing. So this would be one that you'd want to have some skepticism about. Other ones, maybe the effect is even bigger than the initial paper said. Like these anchoring effects here are huge, even compared to the original effect sizes. So this is the type of thing we need to do to be able to know what effects are real and what are not. And I mean, even more than this, so we can do big whole meta-analyses on just single questions rather than looking at a bunch of different questions at once. There are other mini labs projects. There are some that are looking at different types of samples and different types of environments and seeing if those affect findings. There's some that are looking at how, something that you maybe have thought of before as like a SONA participant, does participating before spring break or right after final, or right before finals or right after the semester starts, are there differences in your performance? So does the subject pool at universities change across the semester? And then another one, a really recent one where they looked at an effect that people thought was pretty replicable. They picked the most replicable effect in a relatively controversial area of research called terror management theory. and tried to replicate it. Terror management theory, just as an aside, is the idea that human beings are a unique species in the sense that we are aware of the inevitability of our own deaths, and that is something that brings about terror. And we have various ways of grappling with that realization that we're going to die eventually. And one of the ways that we've adapted to do that is by bringing ourselves closer to things that will outlast us, like culture or a group that means a lot to us, or our family name. So people that are reminded of death often find those things more important, for example. And an effect in one of the first terror management papers that people really have a lot of confidence in, they just did a many labs project and showed that it didn't replicate at all. Nobody could get it to work. So maybe there are things with terror management theory that need to be significantly updated. Something, that is really important. And I just want to emphasize here too that doesn't mean that the people that did this line of work necessarily did things wrong, or it doesn't mean they're bad scientists. And we really need to do a lot of work of separating the scientists from the result. If you're a good scientist, it means you're adhering to all the types of transparent and responsible things I've been talking about. Not that you found the coolest thing in the world, right? So we need to separate these types of notoriety things and prestige things from the job of being a scientist, and not crap all over these people because they, got a finding that we later realize is maybe not replicable. It's very important to separate those things. So because of things like the mini labs projects, these different norms of things being open, there have been some positive changes as a result of these things. Like some journals have changed their policies such that you get priority for having open methods, open data, and pre-registered hypotheses, or you get little badges that are printed on your paper that show that you did one of those three things: open methods, open data, and pre-registration. And I know it seems kind of silly, but it is really a marker that people are recognizing now that's important to try to get. And some people, they look at a paper and it doesn't say the findings were pre-registered, and they just might not even read it because it's such a necessary and relatively easy thing to do at this point. And I know I keep saying there isn't a journal of replication, and as far as I know, that's still technically true, but there are some journals who have had special issues just on replicating things, so at least that's a step in the right direction. The reproducibility project, again, is a bigger project that, again, was started in the wake of those different events in 2011 and the replicability crisis. This project, it took three big journals selected purposefully, and they selected a random year and a random volume from these three journals within a certain range. So they were all from 2008. And they selected 100 studies from these three journals to try to replicate them with either a close or exact replication. They tried to get in touch with the original authors and made the data the materials and data from the replications available so people could compare them to the original studies. This is the point where I would ask you, how many of these things do you think replicated? And you might be overconfident or underconfident. I know I expected it to be higher than this personally. And it was only, actually, sorry, I thought I had it on the slide. It was only about 39%, it was 38% of the studies replicated. It's pretty low, right? So that's another indication that this false positive rate is probably pretty big. But it's also the case to recognize that just because you have one failed replication, that doesn't mean that you write off those studies. So let me go back and use this as an example. Maybe an original study has an effect size right here. and the next study has an effect size that's right here. Those two studies don't tell you everything, right? So a single replication showing that there is no effect doesn't totally negate this study. Those are just two data points that we need to be able to include in a broader set of data about a given effect or a given study. So, I mean, some people saw this and said, all these findings are bullshit, and we need to string up all these authors and never read their work again. Well, no, it's that this is a necessary step in trying to understand these effects. It's a first step in a long journey that we need to do to replicate lots and lots of different things. So again, people are kind of quick to criticize and be, really critical about all these different things, but it's necessary to have a little bit of restraint in that. So more studies might have replicated if they've had more contact with the authors. If you looked at the studies that actually had input from the prior authors and cooperation there, and had more of an insight into their materials and what they did, then more studies replicated. It was closer to 2/3 of the studies that had that benefit. So maybe there's something that some of them missed. Maybe it's just that some of the studies are wrong, or maybe it's some of them are giving low effect sizes in the replications due to chance. It's really hard to say. There's probably just that they're doing different things and that they have different environmental features too when they're doing the study. Sometimes 6 years apart, totally different places, with maybe different demographics. Some of those things matter. But it is important to still recognize that some of the effects, maybe they're wrong. We need to do more research to try to figure that out. Just to wrap up the class, I don't want to, spend way too long talking about this stuff, but I think it's necessary to wrap up at least a couple more general things now that I've maybe shaken your confidence in some of the things that you have heard about and read about in other classes. I want to, kind of double down on some of this first, though, by saying part of science in general is being skeptical. That when you are engaging with anything, you should not just believe it. You should ask questions. You should have some doubt. You should ask why. You should ask how, why people did things they did, how they came to the conclusions they did. Just ask questions. Don't take anything at face value. But I want to make this distinction I've made before. You should be skeptical. You should ask questions. You should wonder. You should be critical. But you shouldn't be cynical. You shouldn't just assume that everything is terrible and no findings are correct and everyone's lying and everyone's wrong and everyone's p-hacking and no one actually hypothesized anything. Like, that doesn't help. Being skeptical and applying these things that not just in this class, but when you are dealing with things you read in the media or talking to family. Like, you have some expertise now about how science should work that you should use and that you should communicate to others, and that you should help you, through the times in your life when you're going to be faced with numbers that, you're going to be able to interpret that other people might not. In order to not be cynical, it's important to remember, too, that science isn't about just saying facts all the time. Science is about learning. Science is about gaining knowledge. If you look back over the history of our scientific knowledge, most of it was wrong. And a lot of the things that we think we know today are wrong. We just don't have the knowledge or the understanding or the tools to know that it's wrong. But it is our best understanding right now, and that is all that we will ever have. So the only way that things progress and get better, and that we get better technology, we get better understanding of ourselves, a better understanding of the world, is by this pursuit of truth. And the only way that we improve it is by being wrong and recognizing when we're wrong and getting better. So this is part of the game. Having things be wrong and finding out the right thing is just how it works. So having some humility, knowing that the best answer we have now, it is what it is. It'll help us to the extent that it can. But pretty much everything we know, probably 100 years from now, people will look back and giggle about the stupid things that we think. And that's just how it works. But we still do get better. Like, we know things now that people didn't before. It's not that we have, too much black bile and that's why we get cancer, right? We know, in some senses, how tumors work. We know that there are bacteria and viruses. It's not just that, someone who has bad juju gets sick, right? So we do know things that help us, even though our understanding of things in general are probably wrong. We're going to learn more about sickness, and we're going to learn more about how everything works. And the only way we can do that is by doing science in a responsible, transparent, open manner. And when you are in your lives reading about things and reading about science, just take a look at these things and think about the things you've learned over this semester and all your other classes. Be skeptical. Don't believe everything you read. Look at the details, like things about sample size and how things were measured and how they were allocated to conditions and what those, if the conditions actually mean anything in terms of the independent variable. Are you aware of other studies showing this same thing, or is it the only one? Is there discrepancy? Are people ideologically or financially motivated to say something? Are people being transparent? These are important things to be aware of, and it's important for us as people who are scientists or might become scientists, or just people in their everyday lives that are engaging with science and consuming it, to advocate for good research and try to make sure that people are being held accountable and responsible for the types of claims they're making and how they are arriving at those claims. All right, I will step down off the soapbox and I'll wrap up this last video. Despite the weirdness of the end of the semester, I really enjoyed this class. I always love teaching this class, even though, some of the material is a little on the dry side. There's a lot to get really excited about too. I hope you found a lot to enjoy in this class, and keep in touch if you have questions about stuff from this class or grad school or getting involved in research or anything like that. I'm happy to hear from each and every one of you. So stay in touch and good luck with everything else. Stay safe.